welcome to this webinar about SARS-CoV-2-specific B cells and therapeutic antibody discovery. So this is the agenda of the meeting. I would like to discuss about the role of monitoring cellular immune response in COVID-19 and also the recombinant SARS-CoV-2 proteins that we offer and how they can be used for the B cell analysis. And finally, I will end with part on the therapeutic antibody discovery. Let's start with the monitoring of cellular immune response. In general, it is important to study the immune response to viral infection. And indeed, all immune cells can have a role in the disease outcome and clearance. Some, like for example, macrophages, might have a negative role in the pathogenesis of diseases such as SARS-CoV-2 infection and COVID-19. And these cells can be studied as target to develop drugs that can be used to treat patients in an acute and severe phase of the disease. Other cells like lymphocytes play instead usually a positive role in the defense against the viruses and also in the mounting of immunological memory, which gives the protection from future infection. Of course, the goals of vaccination against SARS-CoV-2, as for any vaccination, it is to develop an immune response and therefore measuring SARS-CoV-2-specific T cells and B cells in vaccinated individuals is a very important step in the evaluation of the efficacy of COVID-19 vaccines. The most important difference between T cells and B cells is that T cells recognize via T cell receptor antigens bound to the MHC of infected cells. On the contrary, B cells, via B cell receptor, can recognize antigens of full pathogens present in the microenvironment without the need of a presentation of an infection. Once the B cells recognize the specific antigen, they develop into plasma cells. And the B cell receptor is basically an antibody that lately, as plasma cells, is released and secreted in the environment and can eliminate the pathogens in several ways. Since the antibodies eliminate the pathogens directly before the cells are infected, this response is sometimes called sterile immunity, and it is usually the target of vaccinations. How do antibodies protect us? The most simple way an antibody can protect us is via neutralization. In the case of SARS-CoV-2, for example, some antibodies recognize the protein spike on the virus surface and block its binding to the ACE2 protein, preventing therefore the virus to infect the cells of the body. There are, however, other ways that antibody can use to help to eliminate immune response and to kill pathogens or, for example, tumor cells. One example is the activation of the complement or the recruiting of killer immune cells such as macrophages or NK cells. As we will see later, therapeutic antibodies should be able to induce one or more of these mechanisms, and therefore it is important also to test their functionalities. So we've seen how basically a strong antibody response per se should be able to control and clear a viral infection such as SARS-CoV-2 infection. However, it has been observed in several individuals that the level of antibodies in infected individuals go down after several months after infection. And therefore, these individuals are still susceptible to future reinfection with the virus. However, a recent study have shown and proved that B cells instead persist in the blood of convalescents even over 8 months after infection. And therefore, the B cells could help to prevent future infections or help the individual to clear the virus quickly without developing of a severe COVID-19. Both roles of B and T cells are therefore important in SARS-CoV-2 monitoring. For these reasons, at Miltenyi Biotec, we have developed specific tools to study these cells. In the following part of the webinar, I will illustrate the tools for SARS-CoV-2-specific B cell detection. However, for T cells, we also have several products and solutions I can recommend in order to study the T cell response to SARS-CoV-2. You can find more of this information in upcoming webinars and also in our website. So let's look now at how to monitor antigen-specific B cells using the SARS-CoV-2 proteins. So in the past 12 months at Miltenyi Biotec, we have developed several recombinant proteins of the SARS-CoV-2, as you can see here. All proteins are manufactured in a recombinant eukaryotic system with or without a covalently bound biotin. Most of our tools have been developed to recognize B cells specific for the spike and the RBD receptor binding domain. So for the future data I will show you in the next slides, you will see mainly these two proteins used. And also very importantly, we have released, and we are going to release even more in the future, also the mutant variants of RBD and spike. How can you use this protein? The protein can be used to study molecular mechanism of SARS-CoV-2 virus, such as the binding of the spike protein to the native ACE2 or fluorine cleavage assays. They can also be used to detect antibodies in serum or plasma of infected or vaccinated individuals. However, one of the most important applications of the SARS-CoV-2 recombinant proteins is the study of the SARS-CoV-2-specific B cells, and specifically the phenotyping and enrichment of this rare population of cells. And this is what I will show you in the next slides. How does the system work? So first of all, you need to create a so-called tetramers using a simple protocol. One streptavidin conjugated to a fluorophore can bind to 4 biotinylated protein, SARS-CoV-2 protein, by a short incubation at room temperature for 15 minutes. Then the tetramers are added to a cell suspension of B cells. And the B cell specific for the BCR, for the spike or the RBD protein, will bind the tetramers via the BCR receptor. Since the tetramers is conjugated via streptavidin to the fluorochrome, it is then possible to detect these cells and quantify them at the flow cytometer. You can check out the exact protocol for the tetramerization at our website. In all our experiments I will show you in the next slides, we all started from a pre-enriched population of B cells using a releasable system of the label called REALISE. And specifically here we use REALISE CD19 microbit kit, which indeed allows to enrich CD19 positive cells, which are in the blood, the B cells. After the enrichment via magnetic separation, the bits and the labels, so the antibodies on the cell, on the markers, are actually released via the release system, and it's part of the protocol and of the kit. Therefore, what we further use in the experiment are label-free B cells. And this is also very important for two reasons. First, because CD19 is not so highly expressed on B cells and tend to be easily saturated by labels. Therefore, freeing up the marker CD19 from any binding helps a better staining for further downstream application, like flow cytometry. The second reason is that if you want to do enrichment via microbits, you need to get rid of the bits on the B cell so that you can further enrich antigen-specific B cell with a second magnetic labeling. It is also possible to use the same system to enrich the SARS-CoV-2 B cells, also using microbits, as I show you, but also via sorting. So I will show you these two examples later. Also, very importantly, it is also possible to start directly from PBNC without pre-enrichment. I will strongly recommend it because pre-enrichment helps you to have a much better resolution in the flow cytometry and a higher and better results in the cell separation. So very importantly, we have used two types of tetramer for the same protein. So basically the tetramers we are preparing two different fluorochromes conjugation, one in PE and one in PE-Vio 770. In this way, it's possible to detect the antigen-specific B cells by gating on the diagonal of the dot plot. In this way, the unlabeled cells are excluded. Here you can see the gating strategy by excluding dead cells and doublet, and then by gating on the CD19, which are all the B cells, and then further on the scatters, and then on the two labels via the tetramers. Furthermore, our panels also included anti-IgG, IgA, and IgM antibody that allows also to further phenotype the isotypes and also CD27 to distinguish the memory B cells. And also you can see here that we have tested our system in COVID-19 convalescents versus healthy control. And as expected, we saw that the healthy convalescent show a persistence of B cells after 3 to 6 months after infection. And this was much higher statistically compared to the healthy controls. And this proved that our system is actually very specific for SARS-CoV-2. As mentioned, it's also possible to enrich the cells via sorting using the same panel used for analysis. Here we have used the MAX1 title cell sorter because in this way we could really obtain several advantages that I will show you in the next slide. As you see, we obtain a very high purity of SARS-positive B cells. And on the contrary, of course, in healthy control was not possible to enrich the cells because they were really low. Why to use the MAX1 title in these settings? This is a perfect system to sort in safety fragile and rare cells. It is indeed used in COVID-19 study exactly to sort patient material that can be at risk, give risk to the operator. The microchip of the MAX1 title is disposable, is closed, and is aerosol free. And this really helps to avoid carryover or risk of infection of the operator. After sorting with the MAX1 title, we have taken the same cells and then add them in culture. And here we use the B cell expansion kit. That is a specific kit developed to expand polyclonally B cells via CD40 ligand multimer and also IL-4 cytokine. This combination really helped the B cells to expand and then also to start producing antibodies. What we have observed here is that the antigen-specific, so spike-specific B cells in culture expanded nicely after 14 days, and also they were able to produce IgG specific for spike. Basically, after 14 days, we have collected the supernatant and measured with an ELISA-like system the presence of IgG antibodies against the spike. So not only the antigen-specific cell expanded in culture, but they were also able to produce specific antibodies, proving that these cells isolated with our protocol were alive and active and functional and could potentially be used for antibodies generation. However, there is also an alternative to sorting, that is the magnetic enrichment. It is possible to use SARS-CoV-2 B cell magnetic enrichment via using a second labeling with anti-PE microbit and then run the cells through the column. What you collect as positive fraction will be then the antigen-specific B cells. As you can see from the data, in this way you can obtain a very nice 40 times fold enrichment of the population of need. The purity is not as good as with sorting, but it's possible in this way to have a high enrichment of the wished population, which is a very rare population usually, and then you can further do serial dilution to isolate the single cells and single clones. From the 1st of April, it's also possible to collect to buy all these reagents and also to have a clear protocol in the datasheet by buying the specific SARS-CoV-2 B cell kits. We offer a SARS-CoV-2 B cell analysis kit that includes all the panels of antibody I showed you before, and either the spike protein or the RBD protein. Instead, for the magnetic enrichment, we offer SARS-CoV-2 B cell microbit kit that can also be either for spike or RBD, and is composed of streptavidin, the protein, and also anti-P microbits. And now let's step into the therapeutic antibody discovery. So in order to develop therapeutic antibodies, for example, for COVID-19 treatment, but also for cancer treatment, many steps have to be followed. First, you need to define a target and validate it. Then you need to discover which antibody binds this target. Then you have to produce the antibody in a stable system. Once they are produced, you want to test them functionally to make sure that they are optimal for their purpose. For example, as I showed before, if they are neutralizing or they can be used to recruit other cells or opsonization or other function. Very often, antibodies are also engineered to make them more stable or to make them more specific and more able to bind the target with high affinity and so on. And finally, you want to select the right candidate that can be used in clinical testing. So you will have to do some preclinical data. And if then the product goes into the clinical trials, you will have a candidate drug that can eventually, after all the phases of clinical trial, become a final pharmaceutical product. In the second part of the webinar, I will therefore focus on the antibody discovery, but invite you also to follow other webinars available in our Miltenyi Biotec database that explain other steps such as the functional analysis of antibody candidates that you can see here. So let's look now about what is the process for antibody discovery. So what is the good antibody candidate and how do you find it? So ideally, you can assume that subjects exposed to a certain pathogen might be able to develop an immune response to it, including specific antibodies able to eliminate pathogens. And you might want to start from these individuals to collect antibodies that could be effective against the pathogen. In COVID-19, for example, this search was done looking into the serum of convalescent individuals able to neutralize the virus and then recover from the disease. Therefore, most of the approaches to identify antibodies suitable for therapy look for natural antibodies present in the body of human or animals that have been in contact either with the pathogen or with an immunization to that pathogen. However, an unbiased approach is also possible by using libraries of synthetic genes. So the scope of the antibody identification is to determine the sequence of the antibody in order to clone it in an in vitro system able to express the antibody in culture for a mass production. But how do you practically identify the antibody sequence? Antibodies are found in the serum, but it's basically not possible to just isolate the immunoglobulin proteins from the blood and understand the exact sequence just starting from the protein purification, although some advancement has been done in this direction using mass spectrometry. But the only way to properly identify a specific antibody and its sequence is actually to identify the B cells that can produce it, or the plasma cells, and then sequence the genome in order to determine the sequence of the antibodies that they produce. But how can you find and isolate B cells or plasma cells? So let's have a look at the typical development path and compartmentalization of B cells and plasma cells. The B cells develop in several steps. As you can see here, it's quite a complex maturation process. And then they can become either follicular B cells or marginal zone B cells, and they will enter eventually in the lymph node. At this different step of this complex differentiation, they might develop into plasma cells, and the plasma cells are then secreting the antibodies. But where can you find these cells and how can you really isolate them, and which are the one appropriate for therapeutic antibody discovery? So first of all, you need to see where the cells are located. If you want to work with patients, or in material from human, you cannot imagine to collect it from certain tissue because it would not be also ethically allowed. It would be much easier to access the cells in the blood. And as you can see here, a lot of the first step of the B cells maturation happen in the bone marrow. Then the cells goes into the blood, then they go again in the lymph node, so they're again not accessible. And in some cases, they are in blood, like in the case of plasmablasts, but only for a very short window of time, so between 7 and 14 days after immunization or after contact with the infectious agent. And at one point, they become memory B. They are in the blood and they persist also in the blood for long, as we've seen, for example, SARS-CoV-2, but then eventually enter again in the bone marrow to become long-lived plasma cells. Plasmablasts also can be found in the blood, but also in this very narrow window. So it's actually not very easy to find cells, a B cell specific for a certain antigen, unless you really look for memory B cell in the blood. Plasma cells have a very short window in which are present. And also the second problem is that plasma cells, they don't express anymore the PCR on the surface, so they cannot be fished out by using antigens. So that's why most of the people focus on the isolation of memory B cells from blood. Also, after each single step of maturation, B cells improve the binding efficacy to the antigen. Therefore, it's really important to take them at the very end of the development. This illustration that I taken from a very informative review that I mentioned here below, you can see how specific antibodies can be identified starting from memory B cells indeed. The driving idea of these approaches is that the B cells need to be isolated in single clones of identical B cells, all producing the same type of antibodies. Once the clones are then separated, the single step is to identify the ones able to produce antibodies specific for the target antigen, and indeed, ideally, functionally active. Finally, it is necessary to sequence the B-cell clones to identify the antibody sequence and then express it in a different cellular system for mass production. After the first isolation of B cells, there are mainly three ways to identify single clones. On the left here is the sorting of the single cells in separate wells, and then the culture on feeder cells in order to collect supernatant to test the antibodies of each clone. In the middle, you see the other option, that is to fuse immediately the B cells with an EBV in order to immortalize them and then sort the single cells into wells. After immortalization, the B cells will not need feeder cells anymore, and they could be able to replicate and then stay in the well without problems. On the right side, you see another approach, which is the direct sequencing and cloning. That makes sense, for example, if specific antigen is known and all the sorted cells are antigen specific. This is the case of COVID-19, where spike-specific B cell can be sorted to identify neutralizing antibodies against the spike protein. This approach might not require single cell sorting and culture, but simply single cell sequencing, starting from a population of antigen-specific B cells. Here you can see an example of what we offer as product and solution and instrument to support the discovery of therapeutic antibodies. So first, you can start from the preparation of the sample. You can start from blood, but in some cases, like in the case of cancer, you might start from cancer tissue coming either from patient or from animal models. Some users that want to work on human samples, they might wish to start from different type of starting material. And some wish to directly separate the cells from the blood, so in this case, the B cells from blood, and other, they want to further step of PBMC isolation. We have actually solution for every of this solution and also for different type of starting blood material. After the B cell separation, with the respective type of separation systems, the B cells then can be analyzed using flow antibodies and a flow cytometry like the MAX1. Then the B cells can be cultured for expansion, and here also we offer some media. As I showed you before, we have a B cell expansion kit that works very nicely, and also other type of reagents that can be very useful in B cell activation, for example, TLR ligands and cytokines, and also, as mentioned, the CD40 ligand multimer kit. And finally, another important step that could be part of this workflow is the sorting. As I mentioned before, the MAX1 title cell sorter is very ideal in the case of fragile cells such as B cells, because it's very gentle to the cells compared to normal droplet-based sorters. And also in case of infectious material starting from infectious diseases, a patient infected with infectious agents is ideal because it doesn't carry over samples, it doesn't allow aerosol, and has disposable cartridge. And then finally, once the antibody has been identified, as mentioned before, there are many ways that they can be screened. And this will imply then the use also and the isolation of other type of cells, for example, NK cells for an ADCC assay, or dendritic cells or any other type of antigen-presenting cells for an MLR assay. And for that, as I mentioned before, I will invite you to look into our website and our offer for all the webinars for Miltenyi Biotec that can give you more information on how to set up assay for antibody validation. As you see, I mentioned before, we have seen antibody discovery, and also I mentioned briefly about functional testing of the antibodies. And unfortunately, we are now at the end of this session, but I wish you that you could also look into our different material available either in our website, or also if you contact our sales representative to really investigate further which are the options and solutions that we offer for antibodies discovery and validation. And I'm really happy that you have been part of this webinar. Thank you for your attention.